Welcome to the deep dive. We're here to take complex stuff and, well, make it make sense, just for you.
That's the plan.
And today, we're really zoning in. We're tackling the body's building blocks,
Yeah.
epithelium and glands.
Foundational stuff. Absolutely key.
Right. And our mission, yours and ours, is to get these concepts so clear you could honestly turn around and explain them to a super curious 10-year-old. Think of it like your shortcut, your toolkit for really understanding the body's architecture.
Exactly. From the skin you can touch to, the glands deep inside making essential fluids. We're covering it.
We are.
And what's fascinating, I think, is that these might seem basic, right? Epithelium, glands, but they're fundamental. Understanding them helps you understand, well, everything else, how we breathe, how we digest. It really raises that question, doesn't it? How do we make these complex cell structures memorable, explainable, especially if the goal is knowing it well enough to teach someone else? Luckily, we've got some great source material today, a real treasure trove, actually, provided just for you. So let's dive in and bring these tissues to life.
Okay, let's do it. So before we get into the tissues themselves, histology sounds a bit intense. Why do we even bother with histology?
Yeah, it does sound academic, doesn't it? But histology is simply the study of tissues under a microscope. And it's absolutely crucial. I mean, it's how we understand normal function, how organs work.
Right.
And maybe most importantly, it's how we diagnose disease. Think of it like being a detective, maybe.
Okay.
You're looking at these slides, these tissue samples. They're like incredibly detailed blueprints. Or sometimes, like cellular crime scenes, you're figuring out what's going on. Is it normal? Is something wrong?
So how do we even see these tiny cells? They're invisible, right? It can't just be random pink and purple blobs on the slide. There's a method.
Oh, absolutely. There's a definite method. The most common one, the classic, is hematoxylin and eosin, H&E. And it works based on the chemistry inside the cells. So hematoxylin, that's the blue or purple stain. It's basophilic.
Base-loving.
Exactly, base-loving. So it stains acidic things. Think of the nucleus, the cell's control center. It's acidic, so it goes blue. Then you've got eosin. That's pink or red. It's eosinophilic.
Eosin-loving.
Right, and it stains basic stuff like the cytoplasm, all the, the stuffing inside the cell. That goes pink.
So the house analogy, blue paint for the control room, the nucleus, and pink paint for the rest of the rooms, the cytoplasm.
You got it. That gives us our basic color map, lets us see the different parts. Nucleus, cytoplasm, even features of different cell types stand out.
That's a great visual. So H&E is the workhorse, tells us the basics. But are there other ways, more specialized stains maybe, to get more detail?
Definitely. H&E is fundamental, but yeah, we have other tools. There's histochemistry that uses specific chemical reactions to highlight particular things, like a special test to find hidden treasures in the cells. And then there's immunohistochemistry. Very sophisticated, this one. Probably the most used after H&E in clinics.
How does that work?
It uses antibodies, like tiny targeting missiles tagged with a stain. They bind to very specific molecules, antigens in the cell. So it's like sending in a tiny stained bloodhound to sniff out one particular target. Very precise.
Wow. So being able to see like this,
yeah,
it really is a kind of superpower in medicine, understanding what's happening right down at the cell level.
It truly is. It unlocks so much understanding.
Okay, so let's move to the star, epithelium. What is it fundamentally? Why is it so important?
Right. Epithelium. It's one of the 4 basic tissue types, and it is, well, it's everywhere. The core idea, especially if you're explaining it simply, is that epithelium is like the body's versatile wallpaper, and it's skin.
Wallpaper and skin. I like that.
It covers everything. The outside of your body's skin, cornea. The inside surfaces that open to the outside your mouth, your gut, airways, urinary tract. It even lines internal cavities that don't open out, like around your lungs or heart. We call it mesothelium. And inside blood vessels and lymph vessels, that's endothelium.
So it's lining and covering.
And, crucially, it forms your glands. All of them.
Wow. Okay, so it really is the body's interface. Doing a lot of jobs. What are those key jobs?
Exactly. Key jobs. Well, first, protection, a barrier, like a strong wall against invaders. Think skin.
Makes sense.
Then there's selective diffusion and absorption, acting like a smart filter, or maybe a sponge, like in your lungs for gas exchange, or your intestines absorbing nutrients.
Right, filter and sponge.
And as you said, secretions, acting like tiny factories making and releasing important stuff, which leads us to glands, of course.
Okay.
And it also has roles in, reproduction, sensation, lubrication, getting rid of waste. Quite the list.
That's a lot for one tissue type. What makes it so special? How can it do all that? What are its unique features?
Good question. Several things make it unique. First, the cells are packed incredibly tightly. Continuous rows, like bricks in a wall, really tight, form a solid barrier.
Right.
Second, it's avascular.
Meaning?
No direct blood supply, no blood vessels running through the epithelium itself.
Really? How do the cells survive then?
They rely on the tissue underneath the connective tissue. They get oxygen, nutrients, everything by diffusion from the blood vessels in that underlying layer. They're like clever neighbors who don't have their own kitchen but borrow food from next door.
Huh. That's a good way to put it.
Yeah.
But doesn't that make them vulnerable? What if that connective tissue, the neighbor, gets damaged or its blood supply is cut off?
That's a really sharp point. Yes, it highlights a crucial dependency. The health of the epithelium is absolutely tied to the health of that underlying connective tissue. Damage the support system, and you definitely compromise the epithelium above it. Affects repair, function. It's a big factor in disease, absolutely.
Interesting. Okay, so tightly packed, avascular. What else?
They show polarity, meaning they have a definite top and bottom, an apical surface facing the outside, or the lumen, and a basal surface sitting on the foundation, like a house with a clear roof and a foundation, and everything inside is organized for different jobs at the top versus the bottom.
Okay, organized.
Yeah.
And how do they stick together so tightly? You said like bricks.
Yeah.
Needs good mortar, right?
Exactly. They have specialized cell junctions, different kinds of biological glue or fasteners. Some are like Ziploc seals, tight junctions. They stop things leaking between cells. Super important in the gut, for instance.
Right.
Others are like strong spot welds adherence junctions and desmosomes holding cells firmly together, giving structural strength.
Spot welds. Okay.
And some are like tiny communication tunnels between cells, gap junctions. They let small molecules and signals pass really quickly from one cell to the next.
Direct communication.
Yeah. And they even have special anchors, hemiasmosomes, that bolt the basal layer of cells down onto that foundation layer.
And that foundation, the basement membrane, what is that exactly? It's made of cells?
Good question. No, it's a cellular, meaning non-living, no cells. It's a thin sheet separating the epithelium from the connective tissue below. It's like the strong, flexible foundation slab the epithelial house is built on, made mostly of special proteins like collagen.
Okay.
It's also a critical filter, controlling what passes between the epithelium and the connective tissue. And one last really key thing, high renewal rates.
They replace themselves fast.
Incredibly fast. They're like a construction crew that's always busy, rebuilding, refreshing surfaces. Your skin epidermis replaces itself roughly every 28 days. The cells lining your gut, the enterocytes, every 4 to 7 days.
That is rapid turnover. Makes sense for surfaces that get a lot of wear and tear.
Yeah.
But,
Yeah.
like you hinted at with the blood supply thing, there must be a downside to that constant rebuilding,
Right.
a risk.
Absolutely. It's a double-edged sword. Essential for repair, fantastic design, but that constant rapid cell division, well, statistically, it increases the chances for errors, mistakes during replication. And yeah, that's the downside. In clinical practice, you see the connection. This high turnover rate is a big reason why cancers arising from epithelial tissues carcinomas are the most common type of cancer. It's the inherent risk linked to an essential strength.
That's a really important connection to make. Okay, so this is where it gets even more interesting, maybe. Epithelium isn't all the same. How do we tell the different types apart under the microscope? Is there a system?
But there is, yes. And it's quite logical, actually. Think of classifying epithelium like describing a building.
Okay, a building.
First, how many floors does it have? Second, what shape are the rooms on the top floor? And third, does it have any special features like fancy trim?
Floors, top room shape, special features. Got it.
So the 3 criteria are number of layers, shape of the cells in the topmost layer, and any specializations.
Right.
For layers, it's either simple one layer, like a bungalow,
What floor?
or stratified more than one layer, a multi-story building,
Many floors.
or pseudostratified. This one looks like many floors because the nuclei are all at different heights.
Pseudo, like false.
Exactly. Looks stratified, but actually every single cell touches the ground floor, the basement membrane. It's a clever illusion.
Okay, layers. Simple, stratified, pseudostratified, now shapes. Top floor shapes.
Right. We look at the shape of the cells in the most superficial layer. They can be squamous flat, like pancakes or floor tiles.
Squamous flat.
Cuboidal cube-shaped, like dice.
Cuboidal. Square?
Columnar, tall and rectangular, like columns.
Columnar tall.
And then there's transitional, unique shapeshifters, sort of blobby, umbrella-like cells on top, only found in the urinary tract.
The stretchy ones.
The stretchy ones. Like balloon animals, they change shape as the bladder fills and empties.
Cool. And special features.
The main one is keratinization. Adding that tough, waterproof layer of keratin protein makes the cells dead, essentially, but incredibly protective. Like adding armor, we see that on skin.
Okay, so naming convention is layers first, then shape, then special features if they're there.
Yeah.
Simple squamous, stratified, squamous, keratinized.
You've got it. That's the system.
Perfect.
Mm.
Now, beyond just the shape and layers, some cells have extra tools, right, on their surfaces. Little tools for big jobs.
They do. These are mostly on the apical surface, the top. We call them apical modifications. First up, microvilli.
Microvilli.
Tiny finger-like projections, like little bumps sticking out from the cell surface. Their main job, increase surface area massively.
Right.
For absorption, think of it like having tiny hands all over the cell surface, grabbing as many nutrients as possible.
Like in the gut.
Exactly. Small intestine, kidney tubules, they form a brush border under the microscope. They have a core of actin filaments inside, giving them structure.
Okay, microvilli for absorption. What else?
Then there are stereocilia. Now, confusingly, they're not true cilia. They look like really long microvilli.
Longer fingers.
Sort of, yeah. Also have an actin core. They're involved in absorption, too, like in the male reproductive tract, but also have a sensory role in the inner ear, detecting movement.
Interesting. So absorption and sensory.
Mm-hmm. And then we have true cilia. These are different. They're hair-like projections, and crucially, most are motile. They can move.
They move. How?
They have a core made of microtubules arranged in a special way that lets them beat rhythmically, like tiny oars or brooms.
Brooms, so they sweep things along.
Precisely. Think mucociliary clearance in your airways. The cilia beat together to sweep mucus full of trapped dust and germs, up and out.
Wow, so that's how your lungs stay clean.
Largely, yes. You also find motile cilia in fallopian tubes, helping move the egg. And the tail of a sperm, that's a flagellum, which is basically a very long cilium. There are also non-motile cilia that act as sensors.
Fascinating. And keratinization, we mentioned that's a special feature, but it's also kind of a surface thing, right? The armor.
Absolutely. Keratinization is key. That tough protein makes skin waterproof, resistant to damage. Chemicals, rubbing.
The cells fill up with keratin, lose their nucleus, thicken their membrane, become these tough, dead, protective scales on the very top layer. Essential for survival on land, really. Stops us drying out. Found on skin, parts of the mouth.
Okay, got it. Shapes, layers, special tools. So let's put it together. How does someone, like our medical student listener, or even that bright 10-year-old, start recognizing these in the body? Where do we find them? Connect structure to function.
Okay, quick tour. Simple squamous, super thin, flat cells, perfect for quick exchange. Think alveoli in the lungs gas exchange. Lining blood vessels, the endothelium. Thin is key.
Thin for exchange, right.
Stratified squamous. Yeah. Many layers tough. For protection against abrasion, keratinized on your skin, that tough outer layer. Non-keratinized inside your mouth. Esophagus still protective, but moist.
Protection, multi-layered.
Simple columnar, tall cells. Great for busy jobs, secretion, absorption, lining most of your GI tract, stomach, intestines.
Call them busy. Simple cuboidal. Square cells, often found in ducts, small glands, kidney tubules, good for secretion, absorption, transport, like in pancreatic ducts, a thyroid follicles.
Square for ducts and tubes.
Pseudostratified columnar. Looks layered, but isn't. Often has cilia. Think trachea, those cilia sweeping mucus up. Mucociliary escalator.
The fake layers with brooms.
Exactly. And finally, transitional epithelium. The stretchy superhero, only in the urinary system bladder, ureters. Allows stretch, protects underlying tissues from urine.
Stretchy urinary only. Got it. That really helps picture where they are and what they do. Okay, so from coverings, we move to creators, glands. We said epithelium forms glands. What exactly is a gland?
Fundamentally, a gland is just an epithelial cell, or a group of epithelial cells that are specialized to produce and release something, a secretion.
So specialized factories.
That's a perfect analogy. Tiny factories, each making and delivering a specific product. Sometimes it's just a single cell, like a goblet cell.
A one-person factory?
Pretty much. Makes mucin. Mucin hydrates to form mucus, and mucus does amazing thing. Protects surfaces, traps particles, lubricates, even fights microbes.
Wow, mucus is important.
Very. So those are the factories. Now, how do they deliver their products?
Right, delivery methods. You mentioned some have tubes, some don't.
Exactly. Two main types based on delivery. Exocrine glands have ducts, tubes. They secrete their product onto a surface like sweat glands, or into a cavity or lumen, salivary glands, gut glands, like a factory with a clear delivery pipe.
Exocrine, exit via duct.
Perfect way to remember it. Then you have endocrine glands. They have no ducts. They're ductless.
So how do they deliver?
Directly into the bloodstream. Their products are hormones. So it's like a factory shipping its products straight onto the body's main highway system, the blood, to reach distant targets.
Endocrine, into the blood.
Oh, okay.
Hormones, pituitary, thyroid.
Precisely. Pituitary, thyroid, adrenals, all endocrine.
Okay, exocrine, endocrine. Ducts or no ducts. But even how the cell releases its product, that varies too, right? You mentioned it wasn't just a simple release.
Not at all. 3 main ways cells secrete. Most common is merocrine secretion. The cell makes the product, packages it into little vesicles, and these vesicles just fuse with the cell membrane and release the contents outside.
Like sending out little packages.
Exactly. Clean, efficient, no damage to the cell. Pancreas, salivary glands mostly do this.
Okay.
Then apocrine secretion. Here, the product accumulates at the apical, the top end of the cell, and then that whole top portion of the cell actually pinches off.
Wow, it loses its head.
Sort of. It loses a chunk of itself containing the product. Mammary glands producing milk fat use this. Some sweat glands, too.
We'll book a piece off.
Right. And finally, holocrine secretion. This is the most dramatic. The entire cell fills up with product, and then the whole cell just dies, disintegrates, releases everything.
The whole factory self-destructs.
Ultimate sacrifice. That's how sebaceous glands in your skin release sebum, the oily stuff. The cell is the secretion, basically.
Holocrine, whole cell.
Yeah.
Wow. Merocrine, apocrine, holocrine, different mechanisms.
And what they make also varies. We talk about mucus glands making thick, slimy mucus. They often look pale or foamy on slides because the mucus washes out. And subtilis glands making watery, protein-rich stuff like digestive enzymes. They usually stain darker, more pink red.
Mucus versus serous.
Yeah.
Okay. And they have different shapes too, like the factories themselves.
Yeah, morphology matters too. They can be tubular or rounded like berries, acinar, or coiled. And their duct system can be simple, one duct, or compound branching ducts, like factory layouts, simple workshop, or complex industrial park.
So much variety. Now, sometimes these factories, they go wrong. Cystic fibrosis is a really important example, right? How does that connect to glands?
CFEF is a tragic but really instructive example of glandular malfunction. It's genetic autosomal recessive, caused by mutations in the CFTR gene.
CFTR.
Cystic fibrosis transmembrane regulator. It's a protein that acts as a chloride channel, moving chloride ions. Now, in most glands affected by CF lungs, pancreas, intestines, the faulty CFTR protein means chloride gets trapped inside the cell. This pulls sodium in too, and then water follows by osmosis. So water gets sucked out of the secretions and into the cell.
Leaving the secretions thick.
Exactly. Dehydrated, thick, sticky mucus clogs up the works. Imagine the factory pipes getting gummed up with thick glue instead of smooth-flowing product. That's what happens in the airways, the pancreatic ducts. Devastating effects.
But sweat glands are different in CF.
Paradoxically, yes. In sweat glands, the faulty CFTR means chloride can't get reabsorbed back into the cell from the sweat duct. Sodium stays out with it too.
So the sweat stays salty.
Very salty. Much saltier than normal. And that's why the sweat test measuring chloride in sweat is a key diagnostic test for CF.
Wow. A single gene defect messing up glands in different ways across the body. Powerful example. Okay, to wrap up glands, maybe a quick highlight reel, some specific examples from the sources showing their diversity.
Let's do a quick gallery tour. The pancreas, a real multitasker. It's got endocrine parts, the islets of Langerhans making insulin and glucagon for blood sugar control.
Endocrine.
And exocrine parts, the Acini making digestive enzymes, released via merocrine secretion into ducts leading to the intestine.
Exocrine. 2 jobs in one.
Then the thyroid gland, endocrine, makes T3 and T4 hormones, controlling metabolism. Unique structure. Thyroid follicles, spheres of cells surrounding a storage space filled with colloid, this gel-like stuff where the hormone precursors are stored, like the body's thermostat.
Thermostat with storage tanks.
Good one. Then the adrenal glands, sitting on the kidneys. 2 glands in one, really. An outer cortex with different layers making different steroid hormones, cortisol for stress, aldosterone for salt balance, androgens.
Layer cake of hormones.
Right. And an inner medulla, totally different origin and function, making adrenaline and noradrenaline, the fight-or-flight hormones.
2 distinct factories glued together.
Precisely. Just shows the incredible integration and specialization. We could mention salivary glands too. Parotid is purely serous, sublingual, mostly mucus, submandibular mixed, each making a slightly different recipe of saliva.
Amazing diversity from these tiny structures. What an incredible journey, seriously.
Yeah.
We went from staining cells pink and blue to understanding these intricate coverings, the epithelium, and then these amazing little factories, the glands. I really feel like you, our listener, now have that solid foundation. You get the concepts, the analogies. You could definitely explain this stuff, even to that 10-year-old.
Hopefully. The analogies help, right? Floors, rooms, factories, delivery trucks. Anchors them in your mind.
Totally. Remember, folks, the key is engaging with it. Review the ideas. Maybe try explaining tight junctions or apocrine secretion out loud. It really helps lock it in.
And thinking bigger picture, understanding these basic tissues, it's not just about memorizing names, it's appreciating just the elegance of the design, the body's resilience at this tiny scale, which does lead to that deeper question, doesn't it? How does disruption of these seemingly simple building blocks, a faulty ion channel here, uncontrolled growth there, lead to the huge range of diseases we actually see? How do things go wrong at this fundamental level?
That's a great question to ponder. And if you do want to dig deeper, the sources mention some excellent resources. Weider's Histology, the Histology Guide website. Great places to continue exploring these cellular worlds.
Definitely worth a look if you're keen.
Absolutely. Keep that curiosity firing. Well, that's all the time we have for this deep dive.
It's been great chatting.
Likewise. Join us next time for another exploration. Until then, stay curious.